Hello there, my name is Chris Maycock. I'm a retired IT professional. I live in the southwest England, and I've been diagnosed with Parkinson's about 10 years gone. So I have, as they say, a stake in this conversation. And I'm pleased to introduce Dr. David Beckham.
Hi, I'm an associate professor at the University of Colorado School of Medicine. And I have appointments in the Department of Medicine and Infectious Disease, as well as neurology and Department of Microbiology and Immunology.
David, we really haven't got the time to turn everyone in the audience into virology experts. So I'd like to concentrate as best we can on helping people with Parkinson's, a lay audience, understand the contribution that your science has made and could make to the understanding of our problems. So I'd like us to look first at the structure, understand the structure of the virus, which even with my light reading is clearly very complex. So let's start with what might seem to us to be the easy question. What is a virus made of?
It's a great question. You know, I think the way I think of viruses is they're essentially parasitic genes or genetic elements, and they've basically evolved over since actually first cells were created, prokaryotic cells, bacteria cells, and have co-evolved with these cells over billions of years, and they're extremely well adapted to infect cells, and that's what they do. And so they're made of, in general, basically genes, which are either DNA or RNA, and then they have a few proteins typically that allow them to sneak inside of a cell and cause an infection. But there's a wide, breadth of variety within that simple explanation that allows them to infect just about any cell you can think of. They can infect it. And so they're, like I said, they've been around a lot longer than us, and they're really good at doing what they do.
And the difference in one sentence between RNA and DNA is?
Yeah, so the way I kind of think of DNA is the blueprints that tell you how to build your house, whereas RNA is the actual workers that go out and do all the work. And so some viruses just encode the blueprints and require the cell's workers to do the work. Some viruses actually encode the workers themselves and then sneak into a cell and kind of take it over. So there's a couple of different strategies they use.
Okay, and we'll understand a bit more soon about how they affect us, no doubt. How do you classify them?
So they're broadly categorized, kind of like I was just saying. We have, viruses that infect what we call prokaryotic cells, which kind of typically is like bacteria. You have viruses that infect eukaryotic cells, which is us, and so everything from yeast all the way up to people. And then we kind of take all those different viruses, and we can subdivide them into different classes based on what the genes look like and things like that. And so there's multiple approaches that we use to divide and subdivide these trillions of different species of viruses.
Do we know how many there are, or will we never be able to count them?
Yeah, so so for example, if you just look at seawater, right? There's for every drop of water, there's probably 1 trillion different viruses in that water that are attacking different bacteria in that water. And so that's just seawater. And so you could spend a whole lifetime trying to basically isolate and characterize every different virus in water, soil, and that's not even counting the number of viruses that infect mammals, which is probably a very small portion of the kind of what we call the virome, how many viruses are out there.
I was about to ask you how they get about, but I think I'll leave that question. It sounds as though the answer to that is any which way you can.
Right.
So they are therefore alive, but what does being alive mean to them?
Yeah, so technically they're not alive. Technically they are, because they require a host to basically allow them to reproduce. So they have to get inside of a cell and take over a cell that allows them to reproduce. And that actually disqualifies them from being considered a living organism, if you will. And so, but they are, highly evolved, highly evolved genetic elements, basically, that move from species or, individual to individual.
By definition, from what you say, they can exist for a very long time, waiting to find a new host.
Right, right. Some can persist for quite some time within a cell, waiting, years. Some of the DNA viruses, even in people, for example, the herpes viruses can enter a phase called latency, where they basically hide in the host genome and wait before they reactivate, and then can cause another infection.
So where are their favorite places for hiding, David?
So it depends on, there's multiple different approaches. Some of them can form a little loop of genetic material inside a nucleus or inside of a cell, and kind of just wait and wait to kind of reactivate. Some of them, for example, HIV, actually integrates into the host genes, and can kind of lay in wait in those cells that way. So they have lots of tricks for how they can kind of wait and hide. And then some of them just, like coronavirus, we call them grass fires. They just basically, cause an infection, replicate to high levels, and move on to the next host. So there's multiple different ways they can interact with their hosts.
You must be reading my script, because the next question was what damage do they do? How do they damage the sites that they crash land in?
Yeah, I mean, it's highly variable and a little bit dependent on the virus. Some viruses are highly adapted to people. For example, herpes viruses, a lot of them live in us and we don't even know it. A common one is a virus called human herpes virus 6 that a lot of us get when we're children, and it's in your cells and it's in your body and it's there your whole life, and it replicates sometimes and you never even know it. And it's highly evolved to live within people and to spread, but not make us very sick. And then there's other viruses, tend to be the more recent viruses that jump from species to species. Those tend to be a little more aggressive and make us sick, because they're not as integrated with their host in terms of not making us as sick. And in the end, the only thing the virus wants to do is replicate and spread to another host. And they don't necessarily, quote unquote, want to make you sick, but they do want to use your cells to replicate themselves. And so there's a kind of an ongoing battle for that. So there's a lot of variability in their ability to cause disease.
So in the sense that these viruses evolve, we would say that the ones that have been around the longest are the ones that have learned to live with us, and the youngest are the most aggressive and damaging ones.
That's a nice general rule. There's, in science, as everyone knows, there's an exception to every rule, but it's a nice kind of general rule that the ones that have lived with us for a long period of time tend to try to avoid making us sick, whereas the ones that have recently jumped from hosts, whether it's HIV, coronavirus, Ebola is an example, those can be much more deleterious.
Okay. And the ones that cause the problems, the word inflammation is used a great deal. Is that what they do? They inflame. Could you explain that in a little more detail?
Yeah. So, I mean, in order for them to replicate within a cell, they have to overcome that cell's immune responses. Mm-hmm. And all cells have some type of a basic immune response that prevents, that tries to fight against these viruses. This has been, like I said, an ongoing battle between cells and viruses for billions of years. And so for humans, for example, when a virus infects a cell and it's a newly replicating virus, that cell is going to respond with different immune responses that can result in inflammation. And that inflammation has consequences. It can hurt the cell, it can hurt cells around it, or it can call in other cells from the body that can kind of act like a kind of a cleanup crew. And they sometimes clean up by injuring some of those cells in that area. And so there's some downsides to inflammation, but in terms of the organism itself, the goal is to try to control or get rid of the virus.
What characterizes a virus that can attack the central nervous system? And I think I'm hearing that in principle any virus can get into the central nervous system, and the older ones, the ones that have been around longer, are the ones that are not going to cause a problem. So is that a correct view or not?
Yeah, I mean, I would say, so we have this term called neuroinvasive viruses. So there's probably a subset of human viruses that can actually infect a neuron. If you take some viruses, for example, a GI virus, they may not be able to infect a neuron. So there's very specific viruses that have this neuroinvasive quality or this ability to infect a neuron. Neurons, for some reason, in people or mammals, are quite special in the virus world. There's only a subset of viruses that can actually infect these neurons. And, that's one of the things that our lab's been interested in for a while, is kind of understanding what makes neurons special. Why is it that only a certain number of viruses can infect these neurons? And then how do the neurons respond? So that's one of the things that we're very interested in studying.
One of the things that strikes me about this topic is the difference between viruses that on the one hand live with us and cause no noticeable difference, and those that cause catastrophic damage and bring us down very quickly. Are those in the central nervous system, or can that kind of virus attack any part of the body?
Yeah, I mean, what's also interesting is it's not necessarily mutually exclusive. So, for example, herpes simplex 1 is the virus that causes, the canker sores on your lip or things like that, and it actually lives in what we call the trigeminal ganglia, which are some bundles of nerves up in the face that innervate the face. And 99% of the time when that virus reactivates, it goes out to your lip and it causes a canker sore. For some reason, in some small percentage of cases, it'll reactivate. Instead of going to the lip and causing a canker sore, it'll go to the brain, and it'll infect the brain and cause a terrible encephalitis that's actually can be have really serious consequences. And so there's clearly some interaction between the host and the virus that kind of lead to that event, but it's still a rare event, like you said, even though the virus is living in the neuron.
I want to ask what might be a stupid question. Do you think it is possible that Parkinson's could be transmitted by an arbovirus, by a tick or a midge or a mosquito? Is that
it's a good question. And I mean, so I don't think that what we see as sporadic Parkinson's is transmitted by a mosquito or a tick, or is transmitted by a specific virus. At least in my opinion, I don't think a specific virus is causing Parkinson's disease or sporadic Parkinson's disease.
Good. I'm glad. We can still go down to the woods today, as long as we go separately in our bubbles. Your university and you have been working on this connection between alpha-synuclein and West Nile virus, for example, for many years now. You referred earlier to your work over many years, and certainly your papers, the published ones, I'd see date back to at least 2015. So can you take us through what you have learned in that time, and where you believe we are in understanding the connection between the alpha-synuclein that you see with West Nile and the alpha-synuclein that we see in Parkinson's?
Yeah, it's a great question. So, going back to kind of 2014, 2013, I'm a infectious disease physician as well as a, research scientist. And we were, seeing patients in the hospital, and it was during West Nile season here. We get regular cases here off and on in Colorado. And we were seeing a patient, and the patient had a very focal injury in an area called the substantia nigra of the brain. And she was quite ill. And, we were very interested by that pattern of injury. And we started looking through the literature to kind of figure out, what's different about this substantia nigra, and what could be potentially leading to this injury pattern, or what could be potentially contributing. And we started reading about alpha-synuclein. And alpha-synuclein is clearly related to Parkinson's disease, and has a long history of potentially being an important contributor to the development of Parkinson's. But what we found was people actually didn't really have a great understanding of what alpha-synuclein does in a neuron. It's expressed in a neuron, but what we found interesting is you could knock it out in mice. You could knock the gene out, and the mice don't seem to care. They reproduce normally, they have normal offspring, they function normally. And so we kind of asked the question, well, does synuclein have some role in these virus infections? And so we did what we always do. We just asked the question, and we took the virus that we use multiple different, viruses that like to infect neurons, and we put some of these viruses in these knockout neurons and compared them to just regular neurons. We also did it in the mice themselves. And we found out that without synuclein, the neurons and the mice were extremely susceptible to the virus infection. And so over the years now, we've been working on this question. We've been rather obsessed with it, actually. And we have new data from the lab that suggests that synuclein is playing a role in what we call the innate immune response, the early immune response within the neuron itself to help fight off these viruses. And when you get rid of synuclein, the neurons are much more susceptible to these viruses that infect these neurons. And so what we're hoping to do is to figure out what are the different types of synuclein. Is it the fibrils? Is it the monomeric form? Like, what's the actual, is it the phosphorylated form that we find in Lewy bodies that plays a role? We want to know what's the specific type of synuclein that's playing a role in this kind of immune response to these viruses. And when these viruses infect these cells, does it put these cells at risk for synuclein related toxicity or something like that? And so that's what we've been really interested in, is trying to understand how these links could potentially match up, and could, a virus infection, for example, that your body clears, could that, be one of the risks, if you will, that puts someone at risk, or, one of the triggers or something that could put someone at risk for developing Parkinson's on down the line, especially if, like we said, if you're exposed to multiple different infections over your lifetime, over time, could that add up and lead to an increased risk, with a certain background of genetic susceptibility? Could that increase someone's risk of developing Parkinson's down the line? And so that's kind of the big picture question, I think, that we're obsessed with right now.
Well, certainly I'm obsessed with it, but for an entirely different reason.
Right.
To move your work forward, could you summarize for us the areas in microbiology that need specific attention, and also what and how your animal models need to do in order to move you on?
Yeah. I mean, so right now what we need to do is to understand, how is synuclein potentially functioning within these pathways, within these immune pathways, and what are the things besides viruses that might be triggering synuclein to function in a innate immune pathway. And so, we know that there's other things that can be, that have been linked as a potential trigger to Parkinson's, for example, pesticides and other things like that. So we want to know, is there some common theme here where these triggers are triggering synuclein, and that's making it, in some people form some toxic protein that can hurt the neuron over time, when acutely really all it wants to do in the short term is protect the neuron. But maybe in some people, what it's naturally doing in terms of making an immune response can also potentially lead to long-term consequences. And so we really want to understand those mechanisms. How can we alter them? How could we potentially turn this into a treatment for patients with Parkinson's if we can alter some of these interactions and decrease the risk of synuclein from becoming toxic while it's functioning as an innate immune molecule? Can we alter some of those events and turn, and come up with a treatment? That's kind of the end goal.
How might those treatments be delivered, do you think?
It's a great question. Most likely the way I look at this is if we can find an innate immune trigger that's forcing, if you will, synuclein to form some toxic metabolite or toxic protein, can we block that? So it'd basically be a small molecule kind of inhibitor that would prevent synuclein from interacting with things that would trigger its change into a toxic form. Can we somehow use these innate immune signaling pathways to block that event and prevent synuclein from forming these toxic beta sheet forms that we know are bad, these fibrils that potentially injure neurons? So that's kind of our focus.
I'm sure Simon won't be very surprised to hear me ask the question as to whether you have a position on whether alpha-synuclein is cause or consequence.
It's a great question. I think this is a complex syndrome, Parkinson's disease, and I think there's multiple pathways that lead to toxicity to the neurons. I think synuclein is one of them, and I think it's important. But I think it's pretty clear based on the data that there's other genes that play an important role. And whether synuclein is important in the endpoint in terms of toxicity for all those different syndromes, I would be surprised. I think Parkinson's is a complex syndrome with probably multiple different toxicity events that can lead to a similar kind of what we call phenotype. So I think synuclein can definitely be one of those, but I think there might be others, that contribute as well.
So how can the Parkinson's, people with Parkinson's community help you move on in your work?
I think that's a great question. And since, we were chatting before this, I was thinking about that, and I've thought about that a lot. And I think one of the most important things is just for the people in the Parkinson's community to stay engaged like you are, and to really push the research world to think a little bit outside the box. I think, concepts like this are a little bit outside the box, honestly. And it's in the scientific community tends to be pretty conservative in terms of what they want to work on. And so I think having advocates in the community, pushing for the scientific community to think a little bit outside the box is, honestly, it's probably the most helpful thing from my point of view, is because, our research is obviously a little outside the box.
Are you so far outside the box that you don't collaborate with other research institutes, or are you sharing your work?
That's a great question. No, so we're very collaborative. Actually, one of our best collaborations is there in the UK with, we've been collaborating for a few years now with Thilo Kunath at the University of Edinburgh, and he's been an amazing collaborator. We've been working closely for a few years now too with Patrick Brunden in his laboratory at the Van Andel Institute. And actually just recently, we started up another collaboration with a group at UPenn. I think there's a real interest in the field in asking some of the questions that we're asking, and you know, trying to take this different approach. And so I think we're slowly getting there. It's just sometimes science moves a little slower than you know, in terms of getting there than I'd like. We'd like to move a little bit faster.
Yeah, I had noticed.
Right.
I think on that point, I'd like to thank you very much for the time that you've given and the explanations that you've given. I found it most interesting, and I really am quite excited by what you've had to say. So, David, thank you very much indeed.
Thank you so much. I appreciate your time, and thanks for inviting me.